As we start looking at the process of DNA replication, first thing that we want to recollect is that DNA polymerase, one of our main enzymes, can synthesize only in one direction. We need a 3' free for this to be able to work. One of the other things that we need is a short RNA primer. Because we have the issue of the enzyme only working in one direction, we have two things that are going on, and that's why we call it semi-discontinuous replication. The leading strand is actually synthesized continuously from that single initial primer, but the lagging strand is synthesized discontinuously, and so it has multiple priming events. Each one of these little sections is called an Okazaki fragment. So we're going to go into a little bit more detail, but the first thing I'm going to do is I'm going to have you watch a video. We're going to talk about these again. We're going to give a little bit more detail in a little bit, but I want you to see the process because this is something that I just simply can't do for you.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
That hopefully sets the stage for us. Let's look at some of this in a little bit more detail. So remember what's happening here. We've got the new strands being built by using the complementary nucleotides. Now the helicase, remember, is unwinding, unzipping the DNA helix. And this is a specific motor protein. Again, remember, DNA is found in all life, and this is another enzyme that is found in all life also in some form. We have this partial opening that is called the replication fork, and as helicase moves down, this replication fork also moves down. And it's breaking those hydrogen bonds between those paired nucleotides that we talked about. Remember, that does require energy. Also remember that as we unwind DNA, we're causing torsional strain. So we have another enzyme, topoisomerase, that is going to prevent it from rewinding. We also have these single-strand binding proteins that are coating the newly parted DNA strands. And that's, again, trying to make sure that we keep those strands apart. Now, DNA primase is also found in some form in all life. And this is the thing that makes that DNA primer. We need it to replicate DNA. We literally can't start the replication process without this RNA primer. And what DNA primase does is it uses this single-strand DNA sequence to start the primer. Whenever you see SS in this context with chromosomes or with DNA, that means single strand. If you see DS, it means double strand. Later on, the RNA primer will be removed, then replaced with DNA by DNA polymerase I. Now remember, the lagging strand and leading strands are different. One of them has a 5' end and one of them has a 3' end. Remember, we can only build DNA from the 3' end. So when we look at the leading strand synthesis, it's continuous. It's the one that has the 3' end to start it. So we end up with just this single primer, and then we can start building from there. This strand is extended using that DNA polymerase, and this is, again, always 5' to 3'. So we need that 3' hydroxyl group on the ribose to synthesize the DNA. And then the 5' phosphate group on the next nucleotide or on the nucleotide is going to covalently bond to it. Now the lagging strand synthesis is discontinuous. This is the one that the little video talked about, having to do it in little chunks or little sections. We need an RNA primer for each of those Okazaki fragments. We have DNA polymerase III coming in and synthesizing each of those little fragments until it runs into the previous primer, and that's when it knows to stop. So remember that synthesis of the lagging strand is in the opposite direction. The leading strand is just being built this direction, but the lagging strand has to be built piece by piece by piece by piece, and each one of them has an RNA primer. The RNA primers that are found between the Okazaki fragments will be removed, and then DNA poly 1 will come in and replace it with DNA. And then DNA ligase comes in and seals those backbone fragments together. We see this as a replication fork going in two directions. And remember, a replication fork is formed by the opening of the origin of replication. So if this is our origin, these enzymes can work in both directions. So helicase is going to split in both directions. That's the leading strand as it moves in this direction, and then this is the leading strand as it moves in this direction. We still see the same idea where we've got the RNA primers synthesized by the primase, and then DNA polymerase comes in and fills in those short little stretches, and we make those Okazaki fragments. Primers are removed and replaced with DNA by DNA poly 1, and then the fragments are joined by DNA ligase. The big takeaway here is this particular diagram gives us the idea that it's not just going in one direction. The new thing in this diagram is that it moves in both directions. So eukaryotic replication deals with the idea of multiple replicons. So this is multiple origins where it can start. This increases the speed of replication, and this is particularly important for larger chromosomes. Pause. 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 And now we're going to talk about this idea of telomeres, and this is an interesting topic. Telomeres are a series of short repetitive nucleotide sequences that are found on the ends of all eukaryotic chromosomes. The sequence is TTAGGG over and over again, hundreds, maybe thousands of times. And their job is to protect the ends of the chromosomes from deterioration. It also protects them from fusing with other chromosomes. In addition to that, we're protecting the ends of the chromosomes from nucleases, which are enzymes that cleave the chains of nucleotides into smaller units. The ultimate purpose here is to maintain the integrity of the linear chromosomes. The reason this is important is because for each replication, each chromosome is shortened a small amount every single time that cell divides, the DNA has replicated. And this gradual shortening of the chromosomes has been called the telomere problem. This is the overview of what's actually happening here. Now, the leading strand can be replicated to the end of the chromosome. No problem. We have the primer that goes in, and it gets replaced, and there's no problem. But for the lagging strand, we have that primer that's placed in there to build this back. Primer to build it back. So the issue that happens here is that RNA primer gets removed, but now this can't be filled in because it needs an RNA primer to fill that in. So the end is shortened. And this is the telomere problem. Each replication, we lose a little bit of the end of the chromosome. The good news is that we have telomerase. This is a really important enzyme, helps maintain chromosome integrity. This is where the enzyme itself lengthens the telomere section at the end of the lagging strand. Remember, the leading strand, no problem. Here's our overhang right here. This is the piece that we're not able to replicate because we don't have a place to put our RNA primer. So what telomerase does is it's adding nucleotides to the end of that lagging strand. This is making a section of DNA that now we can use as a template to make that RNA primer that we need. And this helps solve the telomere problem. But as with everything, it's not perfect. It extends the telomeres in certain cells. This is expensive. It requires energy. We don't see this in somatic cells. We see it is in germ cells or stem cells, cells that are going to undergo a lot of division and perhaps give rise to a new organism. Telomerase is developmentally regulated, and we see a definite relationship between increase in age and the deterioration of telomere length. As we get older, we can see here's our telomere, it's getting shorter, shorter, absent, and now we may be working into that chromosome. This, in essence, limits the number of times cells can divide. This is one of those problems that people are trying to solve because this is part of that aging process. Stem cells have high telomerase activity because they replicate often, but sadly enough, cancer cells also generally show high telomerase activity, so they can continue to divide without aging. Pause. 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 So the last big topic we're going to look at is the idea of DNA repair. There are errors in replication. While DNA polymerases are able to proofread, it's not perfect. We also have this additional issue of mutagens. And mutagens are any agent, anything that increases the number of mutations above the background level. There are always mutations with these errors in replication, but mutagens increase it. And we see that particularly in things that we're probably familiar with, radiation like sun damage and chemicals. So DNA repair is important, and there are a few different repair systems that have been discovered. They fall into two general categories. non-specific repair, which is exactly what it sounds like. It's targeting a specific lesion in the DNA, and it only deals with that specific damage. And then we have non-specific repair, which uses a mechanism to repair multiple problems found in the DNA. A specific repair mechanism is photorepair, and it is one particular form of damage caused by ultraviolet light. And this is thymine dimers. The two that are side by side, as you can see here, join together. And you can see it causes an issue. We're losing materials here. So we have photolyase, and photolyase is an enzyme that repairs those thymine dimers. So here we can see they're supposed to be right next to each other, but what ends up happening is instead of being attached to the nucleotide across on the complementary strand of DNA, instead they join together next door to each other, and we get this little pooch here. What photolyase does is sort of ingenious because remember light, ultraviolet light has caused this. Photolyase absorbs light in that visible light range and then uses that energy to cleave the thymine dimer. So the energy that caused it is also being used by the enzyme to fix it. So it absorbs the light and uses the energy. And really what happens is it cleaves out the piece that has the mistake. And then DNA polymerase, remember we talked about that when we were talking about DNA synthesis. DNA polymerase comes in and fills in the now missing pieces. And then DNA ligase, again, an enzyme that we talked about when we were synthesizing DNA, comes in and joins that backbone up. Now, when we look at non-specific repair, we're looking at some damaged region being removed and replaced. There are 3 steps here. The steps themselves are fairly straightforward. Recognize the problem, remove the damage, and resynthesize. So now, as we look at prokaryotic replication, one of the things that's interesting here is this is really quite well studied. It's well studied because the genome is quite small and because there are many mutants, so it gives the opportunity to look into a lot of detail. E. coli has only 4.6 million base pairs. The chromosomes are replicated in about 42 minutes at about 1,000 nucleotides per second with very few errors. So remember, prokaryotes have the single circular molecule of DNA. Replication begins at a single or specific point of origin. It will proceed in both directions. The same process occurs. We've got a leading strand and a lagging strand. All those basic details of how DNA polymerase works, it has to work from 5' to 3', still holds true here. Little difference, the Okazaki fragments tend to be a little bit longer than in eukaryotes, but we still see them. Now, E. coli has 3 DNA polymerase. They've got DNA poly 1 that's acting on the lagging strand to remove the primers and replace with DNA. That should sound familiar to us. We have DNA poly 2 that's actually involved specifically in DNA repair processes. And we've got DNA poly 3, again familiar to us, which is our main replication enzyme. Now, I have this picture here, this diagram here, not for you to memorize it, but for you to understand that this is a very complex enzyme. All 3 of the E. coli polymerases have exonuclease activity. They are able to proofread. Pause. 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 Okay, I want to play one more video for you. And again, this is dealing with our DNA replication. It does start a little earlier, but I think it puts all the pieces together.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.